Welcome back in today's video. Good afternoon, everyone. Today we're going to learn about the cell envelope of gram-negative bacteria. Now, this gram-negative bacteria have the cell envelope which is much more complex than the gram-positive bacteria. Why? Because it has an outer membrane, cell wall, and number 3 is plasma membrane. Okay, so the cell envelope of the gram-negative bacteria has these 3 parts. Now, I'll start with the outer membrane. So basically, in a simplified way, this is an outer membrane. This one is thin PDG layer, the cell wall, and this one is the plasma membrane. Okay, and this one is the outer membrane. So these spaces between the outer membrane and the PDG, and the space between the PDG layer and the plasma membrane, is known as periplasmic space. Okay, so this is a usual structure of a gram-negative bacteria cell envelope. Now, in case of outer membrane, as I have already discussed about the cell wall structure, how the peptidoglycan layer and what are the sugar moieties, what are the amino acids are present in case of gram-negative bacteria in my previous videos. So in this video, I'll focus on the outer membrane structure. In case of the outer membrane structure, there are 2 major constituents. One is Brown's protein, and another one is lipopolysaccharides. Okay, so Brown's protein and lipopolysaccharide. Now, what is the function of this Brown's proteins? The Brown's proteins are basically small lipoproteins. Okay, Brown's proteins. These small proteins are basically embedded in the outer membrane and binds with the PDG layer. So with the help of this Brown's protein, outer membrane, which is basically lies at the outside of the cell envelope, and then at the inner portion the cell wall is present, that is the PDG layer. So there needs a connection between the outer membrane and the cell wall, and this is the major protein which will make this connection between these 2, cell wall and outer membrane. So this is basically a principle which describes about the linkage between the cell wall and the outer membrane. Now, another major constituent, as I have mentioned, that is the lipopolysaccharide. The lipopolysaccharide as it is indicated that there is the presence of lipid plus carbohydrate. Okay, so the presence of both lipid and carbohydrate, that is why it is known as lipopolysaccharide. Now this lipopolysaccharide has total 3 components. What are the components? Number 1 is lipid A. Number 1 is lipid A, which is basically a major component of this lipopolysaccharide. Number 2 is core polysaccharide. Okay, number 2 is core polysaccharide, and number 3 is O side chain. Okay, so these 3 are major constituents of the lipopolysaccharide, and this lipopolysaccharide is the major constituent of what? The outer membrane of gram-negative bacteria. Now, the lipid A contains 2-glucosamine sugar moieties. Okay, 2-glucosamine sugar moieties, and each of the sugar moieties contains 3 fatty acids. It is very important. The lipid A contains 2 sugar moieties, which are basically the glucosamines, and each of the sugar moieties contain 3 fatty acids and phosphates or pyrophosphates. Okay, so this is the structure of lipid A. And the most important thing is this fatty acid component of lipid A, it is basically embedded in the outer membrane, and the rest of the portion like O antigen or O side chain, whatever it is, and along with the core polysaccharide and other proteins, they are extended or projects from the surface of the bacterial cell envelope. So this is the whole structure of lipid A, which is a major constituent of what? The lipopolysaccharide of outer membrane. Now the second one is what? That is the core polysaccharide. Core polysaccharide is basically, again, contains 10 sugar moieties. It contains 10 sugar moieties, and these sugar moieties are very much unusual in their structure. They are not very similar, or they are not symmetric. So these are basically the 10 sugar moieties which is contained by the core polysaccharide, and they are unusual as per their structural analysis. Then finally they have the O antigen, the third one, the O antigen. O antigen is also a polysaccharide. Okay, so this is also a polysaccharide. Now this polysaccharide is basically, this O antigen is basically a polysaccharide which is extended outward from the cell envelope. Okay, and their sugar composition also varies between the bacterial strains. So it's not conserved, the sequences are not conserved. O antigen is conserved, but their constituents are not conserved. So lipopolysaccharide is itself a major constituent of the outer membrane along with the Brown's protein, and the lipid A contains 3 different types of components as I have described here. So thank you for watching this video.